Okay, so what I'm going to be talking about in this video is types of gene mutations, the changes that occur in our DNA and sometimes linked to diseases or genetic variations. Even a single base change can have a big impact on how a protein functions. These are like animals that have mutations. So the first type of mutation we're going to be discussing is a point mutation, which affects just one nucleotide, and there are going to be a few types. So first we have a missense mutation, which changes one amino acid in a protein, and this can alter its function. Now we have a nonsense mutation, which changes a codon into a stop codon, ending its translation early. This usually creates a non-functional protein, like in certain cases of cystic fibrosis. A silent mutation changes a base but doesn't change the amino acid at all, thanks to the redundancy of the genetic code. Point mutations can also be grouped as transitions, when a purine replaces a purine or a pyrimidine replaces a pyrimidine. And transversions are when a purine and a pyrimidine switch places. Deletions and insertions, also called DIPs, occur during replication and repair. These can add or remove one or a few bases from the DNA sequence. When this shifts the reading frame, it's called a frame shift mutation, which can completely change the amino acid sequence. There are some examples of this in breast cancer. Another type of mutation is the simple sequence repeats, or SSRs. These are short DNA sequences that repeat over and over again. When the numbers of repeats expands, it can lead to disorders like Huntington's disease. Finally, there are copy number variants, or CNVs. These mutations involve large sections of DNA being duplicated or deleted. CNVs can contribute to genetic diversity, but sometimes they're linked to diseases. So overall, gene mutations come in many different forms, from a single base change to a large DNA rearrangement. They can be harmful, harmless, or beneficial, but they're a major reason to why our DNA is different and unique.